Hello. Hopefully this is the final lecture that I'm re-recording before exam 2 based on the original file having some technical issues. Again, I apologize if this does not match your notes exactly. And I also apologize for the lack of student interaction here. I do want to make sure that we get this file uploaded so that the students have it available for exam preparation. At this point in the semester, we've been talking about T cell responses in the periphery. We've seen how T cells are activated and do signal transduction through their T cell receptor, as well as signal 2 and some cytokine signaling. We've seen IL-2's role in that activation and in clonal expansion. And we've looked a bit at the differentiation, particularly of CD8 T cells, where they go from being a CD8 positive T cell to becoming an effector CTL that can perform cytotoxicity and kill some infected cell in the periphery. That's important that we see killing in the periphery and not at the beginning of this process because we don't want the CD8 T cell to kill the APC upon first recognizing antigen. We want our cell to sort of acquire that ability when it gets to when it's been activated, when it gets to the periphery, in order to act on an infected cell. And we left off last time talking about the ways that CTLs are able to be cytotoxic. One is that CTLs will have the protein Fas ligand on their surface. Fas ligand will be able to interact with the protein Fas, leading to apoptosis in the infected cell. There was a question last time about how the CTL chose which method to use to kill the infected cell, and in a lot of ways you can imagine it that the CTL is probably trying all of these methods, but you might imagine that some virus-infected cells do not express Fas, in which case having Fas ligand wouldn't do much for the CTL. The CTL can also release granules full of cytotoxic components. Those include perforin and granzymes, and the effects of those were discussed last time. In addition, CTLs often produce interferon gamma as part of their activity, and frequently when we're measuring CTL activity, we might measure interferon gamma production by those CTLs. But the piece of this that we still need to talk about is the differentiation part for CD4 T cells and the effector function of CD4 T cells. If I haven't previously defined it, effector function is just the ability of the cell to have an effect and do something about infection. Here, that signal 3, the role of the cytokine that acts on the naive T cell coming from the antigen presenting cell when activating that T cell is going to be very, very important. That's going to be a big part of what we see our CD4 T cell needing in terms of its activation. When I talked about this slide previously, I didn't really tell you what the effector function of a CD4 T cell is. With a CD8 T cell, I always talk about that cell as being a CTL when it's an effector cell and killing cells. The effector type of a CD4 cell is known as a helper cell. And it's a little bit harder to explain exactly what that cell does. The easiest way to say it is that that cell makes cytokines to influence other cell types. But the exact influence and the exact cytokine can vary, and that variation is partially based on which cytokine the naive T cell received when it was first activated. So helper T cells, which are the effector type of CD4 positive T cells, can be divided into many types of subsets. This figure from your textbook shows some of the subsets that are currently recognized in the field, although it is not all of them. I am not going to talk about all of these different subsets, only some of them. These subsets vary based on which cytokine the naive CD4 T cell receives when it is activated. That cytokine here is listed as the polarizing cytokine. And each of these types of helper T cell effector subsets will make a different type of cytokine and will have a different effector function, combat different types of pathogens, and things like that. Sometimes immunologists really like to come up with a lot of different helper T cell subsets. And so there are a few criteria that seem to be in place in order to sort of define a helper T cell subset as a real helper T cell subset. All of these helper T cells, as I mentioned, function by secreting cytokines. So when those T cells interact with some other cell type, in this case you see one type of T cell in blue interacting with a B cell, that T cell is going to perform directional secretion and have cytokines that are actually preformed in the cell in granules that can be secreted directly towards the other cell to influence the activity of the other cell. We will have things like our adhesion molecules on either side of that region of secretion to sort of keep the cytokines in check. And this helping synapse, I don't know exactly what you would call it, is very similar to the cytotoxic synapse that we saw last time with the CTL. As I was mentioning a second ago, we can come up with a lot of different types of helper T cell subsets. And really, in order to distinguish a helper T cell subset as being sort of a legitimate helper T cell subset, there are three main things that we have to have described. One of those is what cytokine the naive CD4 T cell receives as a polarizing cytokine that turns it from a naive T cell into a particular type of helper CD4 cell. That cytokine often comes from the original antigen presenting cell, and that is often based on which PRR was recognized when that antigen presenting cell saw a pathogen, leading to a signaling cascade and the production of some specific cytokine. Sometimes there might be some other situations where other cells make the cytokine, but for right now we'll imagine it as coming from that original APC. When the naive CD4 T cell receives that polarizing cytokine, there will be signaling through a cytokine receptor that will lead to some changes in the cell. In particular, that cell will now turn on some unique transcription factor. So each helper T cell subset that is well recognized has its own known transcription factor, which is listed here as a master gene regulator. And that master gene regulator or transcription factor turns on all of the genes that CD4 T cell needs to become the particular subset of CD4 T cell that cell is going to grow up to be. And that master gene regulator leads to the transcription of a whole bunch of things in that helper T cell. One of those things would be the effector cytokines that helper T cell subset makes in order to do its job. So in order to talk about a helper T cell subset, it's important to be able to name both what cytokine polarizes the naive CD4 T cell to turn into that subset, what master gene regulator or transcription factor is made by that type of cell, and what effector cytokine is made by that type of cell in order to have some kind of effect. Here you can see an example of how that original APC might bias a T cell, a naive T cell that it interacts with, to become a different type of helper T cell subset. So all of this is really dependent on the innate immune response. Some different PRR might lead to a signaling cascade that leads to a unique type of cytokine. And for example, bacteria might lead to one type of signaling through one type of toll-like receptor or other PRR. That might lead to one type of cytokine, and that cytokine could induce a naive CD4 T cell to become a type of helper T cell that's really good at combating bacteria. So this all ends up being linked together. Whereas a PRR recognizing a fungus would lead to a different signaling cascade that would lead to a different cytokine being produced, and we would activate a different helper T cell subset, one that was really good at combating fungal infections. Sometimes people talk about helper T cells as being something like the quarterback of the immune system. They indicate which direction the immune response will go in based on the types of cytokines they are making. In that analogy, the PRR and that original antigen presenting cell are kind of like the coaches that tell the quarterback which direction it should be sending the rest of the response in. When this whole idea of helper T cell subsets was first described, we saw a description of two types of helper T cells. One was called Th1 and the other was called Th2. For a while, people thought helper T cells, CD4 T cells, could either be Th1 or Th2. We now know that there are a lot more choices. It's not just either or. But this whole process was originally described first for thinking about Th1 and Th2 cells, which are described here. So with a Th1 cell, our naive T cell will receive a polarizing cytokine. Usually the polarizing cytokine for Th1 cells is called IL-12. What you might notice here is that there are particular cytokines that I am going to highlight as being sort of the famous and key cytokines, but your textbook lists multiple different cytokines at different places. On an exam, if you were to list any of the cytokines that were listed by your textbook for a particular situation, I would give you credit. But usually when I think about a Th1 cell becoming polarized, I think about IL-12 as the polarizing cytokine. Once a naive T cell receives some IL-12, it will start making the transcription factor or master gene regulator called TBAT. When our cell starts making TBAT, it will become a Th1 cell, and as a Th1 cell, that cell is going to produce a lot of interferon gamma. And the interferon gamma is going to allow that Th1 cell to make its effector function. One thing that you also might notice is that interferon gamma is listed here as a polarizing cytokine, and so once we start making some interferon gamma, we can further push more cells in sort of a positive feedback kind of way into becoming Th1 cells. So you might wonder what kind of activity interferon gamma is good for. This previous slide lists a bunch of different potential options. One that's important is it does help CD8 T cell activation. It does help enhance APCs. As we'll learn later, it helps enhance NK cell activation. But it is also really important for protecting against intracellular pathogens. And usually when people think about a Th1 response, we think about combating intracellular pathogens. And that is because interferon gamma, when made from one of these cells, is able to bind to an interferon gamma receptor on a macrophage and activate that macrophage. Macrophages, of course, are phagocytic cells that will have internalized some type of microbe. And they may be able to degrade that microbe with many of the effects we talked about earlier in the semester. However, interferon gamma activates that macrophage, and that improves the macrophage's ability to destroy things in its phagosome. It makes the macrophage better at making reactive oxygen species. It makes the macrophage much better at lots of different ways of killing those intracellular bacteria. You can also see that once that macrophage is activated, it will express more MHC, it will have more other types of proteins for stimulating other cells, and so that macrophage is going to get rid of anything it has phagocytosed very well because of Th1 cells. There are a number of different pathogens that are particularly important or that are really known to be controlled by Th1 cells. One of those types of pathogens is in the genus Mycobacteria, which includes Mycobacterium tuberculosis. If you have ever had to have a TB test, you know that there is a skin test that's done, and that's something that we'll talk about a little bit later when we talk about allergy. However, if you test positive on that test, you might be a little bit confused. You might say, well, I, I should know if I have tuberculosis. Um, how could I possibly be positive and not display symptoms of tuberculosis? And the reason is because your immune system sometimes can control tuberculosis with a Th1 response in this structure known as a granuloma. So you are able to actually have Th1 cells and activated macrophages basically surround bacteria in the lungs in order to kind of wall them off and keep the infection contained. And so that person will not feel sick, will not show signs of tuberculosis because the Th1 cells are doing such a good job of controlling that mycobacterial infection. But if those Th1 cells eventually stop being able to control, as they probably will, or if something happens to that granuloma, suddenly the tuberculosis can come back and this person can become infectious. What you may know is if you test positive in the skin test for tuberculosis, you next have to do a chest x-ray. And in the chest x-ray, they are actually looking to see if you have granulomas present, which means that you might later have active tuberculosis. Right now, if you have a granuloma, that means your Th1 cells are doing a good job of controlling the mycobacterium, but later on, you might be able to spread mycobacteria to others. You can also see the important role of the interferon gamma that is made by Th1 cells in controlling mycobacteria here. In this experiment, scientists infected mice with mycobacteria. Here it was not Mycobacterium tuberculosis, this species that causes tuberculosis. It is another mycobacterial species, although we don't need to focus on that too much right now. And you can see that if mice are infected, and they are wild-type mice, which are shown in black, they all survive after they've been infected with this mycobacteria for longer than 60 days. If those mice are missing interferon gamma, you see that they rapidly will die of this infection with mycobacteria. And this is because they're not able to control that infection because they do not have a good Th1 response to that infection. At the same time, another subset of helper T cells was described, known as the Th2 cell. The Th2 cell is polarized when a naive T cell receives the cytokine IL-4. IL-4 leads to that naive T cell starting to make the transcription factor GATA3, which is the master gene regulator in this case. And GATA3 leads that cell to start transcribing effector cytokines. The most important one that I'll mention here is IL-4, although it does seem as though IL-4, IL-5, and IL-13 are sort of all controlled together and tend to be transcribed sort of together in a group. And this will lead that helper T cell to have different types of functions, different abilities to help a different set of immune cells. You can see that IL-4 will lead to a nice positive feedback system with Th2 cells, although you might be curious about where that first IL-4 came from, if Th2 cells are what makes IL-4. And that has been a bit of a source of confusion. It's possible that there are some other cells making IL-4. It's also possible that there is a different polarizing cytokine that we don't fully understand. Some people in the literature talk about IL-33 as potentially being a polarizing cytokine here. So our Th1 cell that we talked about before was really good at protecting against intracellular bacteria. The Th2 cell by contrast, is going to be really important for protecting against extracellular pathogens. So the cytokines made by Th2 cells are going to help B cells with antibody production. I'm not going to spend a ton of time talking about this because as our knowledge of different types of T cell subsets has increased, we've realized that that's not as much of a special Th2 thing. However, Th2 cells can lead to some specific class switch to things like IgE and potentially lead to mast cell degranulation. We can also see things like mucus secretion and peristalsis and eosinophil activation as the result of some of the cytokines that we see coming from Th2 cells. And so the really big thing that we often think about with Th2 cells is mast cell activation, which happens as the result of IgE production. You can see a large repetitive antigen here. It's not drawn to scale, and ideally we'd really like to see it to scale in this situation. That antigen is probably too large to be phagocytosed and is going to be extracellular. And so when our mast cell is activated, it will release granules full of toxic substances, the most famous of which is histamine, that can try to kill that extracellular pathogen. Many of these responses are important in the classical allergy responses as well. And so here you can see a general overview of Th1 and Th2 control, where our original APC makes either IL-12 or perhaps IL-4, leads to either Tbet or GATA3 production, and that turns our cell into a Th1 or Th2 cell respectively. The Th1 cell will make interferon gamma and deal with intracellular pathogens. Good news, an intracellular pathogen induced that IL-12 in the first place. So we've now made a response that will do a good job at getting rid of intracellular pathogens. An extracellular pathogen probably made that first IL-4 burst happen in the case of Th2s, and now we're making cytokines that help to combat the extracellular pathogen. Different cytokines that are made by different subsets of CD4 positive T cells can alter which isotype our B cells will switch to. So we sometimes do talk about certain subsets as being important for B cells, but what you can see is that the IL-4, for example, that comes from a Th2 cell might induce a B cell to make IgG1 or IgE, whereas interferon gamma coming from a Th1 cell might induce IgG3 and IgG2A production. And so again, we're sort of seeing cytokine production impacting the general process and the general set of immune responses we see. Remember that different antibody isotypes will be better or worse at dealing with different types of pathogens. And so here again, we're sort of customizing the response to make a response that is correct for the pathogen that we've seen based on the innate immune recognition. One other really critical thing that we know about Th1 and Th2 cells is shown here. So not only does IL-12 turn on Tbet, which will turn on interferon gamma and promote a Th1 response, this process of turning on Tbet also inhibits GATA3 and inhibits the production of IL-4 and inhibits the production of IL-5. So, and similarly, IL-4 and producing GATA3 inhibits Tbet and inhibits interferon gamma. So production of Th1 responses will lead to positive feedback and the production of more Th1 responses, as well as negative feedback and the turning off of Th2 responses. And the same will happen for Th2 responses. And so we tend to see sometimes a Th1 or Th2 overall bias in an immune response based on cytokine production. There's also some importance between sort of balance between these two sets of signals. Here you can start to see one thing that could be really important here. This is an infection with a particular type of microorganism, still in that genus Mycobacterium, Mycobacterium leprae, the causative agent of leprosy. For a long time, there were two general disease states that had been seen following infection with Mycobacterium leprae. One was known as tuberculoid leprosy, and the other was known as lepromatous leprosy. In tuberculoid leprosy, we could see that the organism was present at very low levels. People who had this were not terribly infectious. There was peripheral nerve damage and granulomas, as well as a little bit of local inflammation. Whereas in patients with lepromatous leprosy, there was a lot of growth of the mycobacterium. These people were quite infectious, and they had infection throughout their body and could see things like bone cartilage and nerve damage throughout the body. And this is the same microbe leading to these two very different disease states. When immune responses in the patients were examined, it was seen that in tuberculoid leprosy patients, cytokines that are made in a Th1 response were observed, whereas in the lepromatous leprosy patients, cytokines that are associated with a Th2 response were observed. And so, in fact, the difference between these two groups of patients was whether they made a Th1 response to the microbe or a Th2 response to the microbe. This could be due to other factors, other infections going on in the patient, patient genetics. It could be due to a lot of different things. Similarly, here you can see that if we were to infect Balbc mice with a parasite called Leishmania, and the Balbc mice are in yellow, those Balbc mice will die as a result of that parasite infection. If we were to give those mice a Leishmania infection as well as an antibody that blocks IL-4, so these mice cannot make a Th2 response, these mice are shown in green, you see that they now are able to survive the infection. So in fact, what this tells us is a Th2 response here is detrimental. The Th2 response is killing the mouse. And if the mouse was able to make a Th1 response, which happens here when we inhibit the Th2 response, the mouse lives perfectly fine. We do know that Balbc mice tend to predominantly make Th2 responses due to some aspects of their unique genetics. So again, you can see here that whether one makes a Th1 or Th2 response to a particular pathogen can influence what happens to you after infection with that pathogen. There's sort of an appropriate response and an inappropriate response to the pathogen. Here you can see the same types of things and examples of what could happen if a person made a Th1 response to a Helminth, which is a type of parasitic worm infection, or a Th2 response to a Helminth. On the left, you see what happens if a person makes a Th2 response. They have IL-13, they make mucus, they try to expel that pathogen, they are making eosinophils, they have IgE, they're activating mast cells. All of those things are going to help protect them against the parasite and maybe clear that parasite. If that person were to make a Th1 response to the Helminth instead, they wouldn't be able to clear the parasite. They would be activating macrophages. Those macrophages wouldn't help against the Helminth, so the Helminth would stick around. And in addition, we would see things like extra inflammatory response, too much inflammation, that interferon gamma might actually help out the parasite. We would see class switch to some antibodies that are completely ineffective, and so the host would end up having a damaging immune response because there has been a Th2 response instead of the appropriate because there has been a Th1 response instead of the appropriate Th2 response. Th2 responses are also particularly problematic in the case of asthma. On the far left-hand side, you can see the lungs in a mouse that has TBET, which means that that mouse is able to make a Th1 response. In the middle, you see airways of a mouse that is deficient in TBET. That mouse cannot make a Th1 response, which also means that it makes an excess Th2 response. And now you can see that that mouse has airway inflammation and airway remodeling, which are some of the characteristic features of asthma. And that mouse may end up getting to things like having excess mucus production in the airway and extreme inflammation where lots and lots of leukocytes have gone into the lung. You can see examples of those on the right-hand side because that mouse is making an excess Th2 response, which we know is associated with asthma. Some of the pathogens that are frequently associated with Th2 infections are different types of helminths. These include roundworms, flukes, and tapeworms. Things like ascariasis, which I showed you on the first day of class, these big worms in someone's hand, as well as things like elephantiasis, which I first saw in my first year biology textbook, and I was so disturbed by the picture, I've never wanted to look at a picture of that disease again. Things like river blindness, which William Campbell in our RISE department won the Nobel Prize for, as well as things like flukes and tapeworms. Schistosomiasis is a pretty famous example. As you look at these parasites, you might realize that they don't tend to be things you think about particularly often in your everyday life. Many of these Th2 pathogens are not things we spend a lot of time worrying about in the developed world. This is what we would see happening with our Th2 response actually doing something to Helminth infection. And I point this out because oftentimes people start to hear about Th2 responses and they're linked to things like allergy and asthma and say, man, Th2 responses are terrible. Th2 responses are dumb and we shouldn't have them. But this is a good version of a Th2 response. You can see that our mast cell shown on the upper left-hand part of this slide has a whole bunch of granules. Those are full of toxic mediators like histamine. If that mast cell becomes activated, it will secrete and release all of those granules. And we might see an example like you see on the right-hand side where we have some type of parasite. Here you see a schistosome larva. So this is actually relatively small in terms of parasites, but you can compare it to the small circles labeled as E, which are eosinophils, and you can realize that those eosinophils would have no way of doing phagocytosis of this very large larva. You can imagine a fully grown parasite would be an even bigger problem. And so because we are never going to be able to phagocytose this extracellular pathogen, the secretion of these toxic products is going to be the best bet to kill this. However, if we actually look at where people tend to suffer from Helminth infection, we tend to see the Helminth infections in the places marked in red on the right-hand side of this figure, largely in places in the developing world. And if we compare this with places where we tend to see high incidence of autoimmune disorders, we see that there's almost a complete switch. And these types of data have led people to come up with something called the hygiene hypothesis. Sometimes you will hear weird takes on the hygiene hypothesis in sort of the popular press, but it does come from some good immunological data that has to do with Th1, Th2 balance. The idea is that in the developed world, we have largely gotten rid of Th2 dependent pathogens while keeping other types of pathogens around. As a result, we have a problem in the balance between our Th1 and Th2 responses and have begun to make some inappropriate Th2 responses, leading to high levels of allergies and asthma. And much of these data come from epidemiological sources. You can see that the concept is sort of shown on the right, that in childhood, the development of the immune system, because of the lack of Th2 pathogens, is sort of held back, and so we see this fight of imaginary Th2 infections and this range of allergic diseases. And we do see good epidemiological evidence that people in westernized countries tend to have more allergy, like asthma, eczema, and other types of allergic diseases, while people in developing countries have less allergies. People in from small families tend to have more allergies compared to those from larger families. The idea being that in the smaller family, you probably might have a cleaner house. You can also see big differences in urban versus rural homes and due to affluence. Homes where there are livestock, so kids who grow up on farms, tend to have fewer allergies. And so we can see these types of associations. Some similar associations are shown here. Children who are only children tend to have more allergies and asthma. Again, the hypothesis is that they are in a cleaner house, whereas those who have multiple siblings tend to have fewer allergies. The oldest child in the family tends to have more allergies, presumably because the house was a little cleaner than when their icky brothers and sisters were around. Kids who go to daycare tend to have less allergies than kids who didn't go to daycare, presumably because they're exposed to more pathogens. And of course, those who grew up on a farm tend to have fewer allergies. Obviously, this is all epidemiological data, and some of the mechanism has been challenging to prove, but we do think it's about balance between Th1 and Th2 responses, and some have indicated that perhaps a good treatment for Th2 responses would be to reintroduce some Th2 pathogens to reestablish the balance between Th1 and Th2 responses. There have been some trials where schistosome eggs that have been killed but will still have antigen have been delivered to try to reduce allergies by giving people appropriate Th2 responses. There are other hypotheses about what the mechanism could be for the hygiene hypothesis, and one of those mechanisms is shown here. So you can imagine that within the population there is sort of a normal distribution in terms of strength of the immune system. Some people have weak immune systems, some people have strong immune systems, and some people have immune systems in the middle. If the population as a whole are infected with parasites, that might shift the entire bell curve, the entire normal distribution, down to a lower strength of the immune system. Their parasites in general may be suppressing everyone's immune system. What that means is that relatively few people will have a super high immune response. And so maybe it's that super high immune response that's actually the problem that's leading to allergies. If we get rid of the parasites in a population, that will shift everyone's immune responses up. The bell curve will be towards the stronger immune system, and more people will have a very strong immune system that might lead them to having allergic responses. And so I just wanted you to realize that there are a few different mechanisms that have been proposed here. Since the original helper T cell subsets were described, some additional types of helper T cell subsets have been talked about as well. The next one that I want to tell you about is one that's called a Th17 cell. Students sometimes get a little bit annoyed about Th17 cells because they think that immunologists have forgotten how to count, going from 1 to 2 to 17. But the reason why Th17 cells are called Th17 cells is that their cytokine that they make, their classic cytokine, is IL-17. So they were named Th17 cells for the IL-17. A cell can become a Th17 cell when it receives a combination of cytokines, and the combination is important. We can see either TGF-beta and IL-6 or potentially TGF-beta and IL-23. As I'll mention in a second, IL-23 was really important in the discovery of Th17 cells. When the naive T cell receives that combination, say, of TGF-beta and IL-6, and note for exam purposes, in this case, you would need to write both TGF-beta and IL-6. You can't just write TGF-beta for this one. That cell will start making a transcription factor called ROR gamma T. Once our cell is making the transcription factor ROR gamma T, it will begin making effector cytokines like IL-17. IL-17 turns out to be really important in turning on neutrophils, and we now know that Th17 responses are really important for protecting against fungal infection as well as extracellular bacterial infection. There are a number of different autoimmune diseases that were originally tied to a Th1 response. That was because of experiments that were done blocking IL-12. We since realized that IL-12 has a cytokine that's quite similar to it, which is IL-23, and in some cases those studies were blocking IL-23 as well as IL-12. And so when people went back and did those experiments more clearly, they realized that those autoimmune diseases were actually linked to IL-23 and Th17 cells, not to Th1. And so there's a lot of evidence of Th17 cells playing an important role in things like multiple sclerosis and another number of different autoimmune diseases. Another important helper T cell subset that has been described is called the TFH, or T follicular helper cell. Again, this one can be a little obnoxious because you might say, oh my gosh, we've even gotten away from numbers now, what's going on? But this one has a name that tells you about what this cell does. So the T follicular helper cell specifically acts in B cell follicles in order to help B cells. So the FH, follicular helper, is telling you about the location where this cell acts. A cell becomes a T follicular helper cell when it receives cytokines like IL-6 and IL-21, and these will result in that T cell making the master regulator, the transcription factor BCL6. Once that cell starts making BCL6, it will produce effector cytokines like IL-4 and IL-21 that will help B cells in their functions in the B cell follicle. And so these will be very important cells in helping to make a good B cell response. We'll talk about B cell responses in the periphery a little bit later. The final set of helper T cell, the final subset of helper T cells that I want to talk about are called regulatory T cells. These were actually the third subset described. For a little while, they were called Th3s, but because of some other details, they ended up getting their own name, which is called Tregs. So what you can see here are Tregs that are being polarized from naive CD4 T cells. Those Tregs are going to receive just TGF-beta. In the previous case, you saw Th17s that were receiving TGF-beta plus IL-6. If you remember, IL-6 was one of our inflammatory cytokines we saw during an inflammatory response. So there, to make a Th17 cell, the cell had to get TGF-beta plus sort of inflammation. Here, our regulatory T cell is getting TGF-beta alone without the additional inflammation stimulus. And when that cell receives TGF-beta, it starts making the master transcription factor, the master regulator, FOXP3. As a result, the cell will start making cytokines IL-10 and TGF-beta. And these cells, these Tregs or regulatory T cells, have the function that they can suppress the immune responses or turn off other cell types. We can see inhibition of other CD4 cells as well as inhibition of CD8 cells, B cells, and many other types of immune cells. In the past, I'll mention a little bit more about this in the next lecture, there were a set of cells that were described as suppressor T cells that had a function like this described, and then there were some data that sort of made suppressor T cells look sketchy, and everyone sort of denied they ever believed in suppressor T cells, and sort of the word suppressor and suppression became a bad word. So typically if you hear immunologists talk about these cells regulate or hear about something being regulatory, that often means turn off or suppress. And so regulatory T cells can turn off other cell types. This may be because of these inhibitory cytokines like IL-10 and TGF-beta, although there are a few different ways that regulatory T cells can act. And you can imagine the importance of these in protecting against autoreactivity and autoimmunity, as well as protecting against a lot of pathologies that we may see with the immune system. Regulatory T cells may act to suppress other cytokines in a number of ways. They may produce inhibitory cytokines like TGF-beta. They also may steal all the cytokines like IL-2 from other T cells by serving as a cytokine sink. And we'll talk a little bit more about in the next class as well. Or they may suppress in other ways. And I'm going to be mentioning this again in the next lecture a bit. Because I had run out of time when I originally gave this lecture. But the last thing I want to point out here is that the reason why things are a little bit more complicated with regulatory T cells is that we have actually realized that there are two ways that a T cell can become a regulatory T cell. This process of receiving TGF-beta is only one way to make a regulatory T cell, and I'm going to start out the next lecture by telling you about the other way to make a regulatory T cell.